The purpose of this video is to supply a little bit more information and guidance for the unknown bacterium lab report that you're beginning this week and is due next week. So I thought the best way to show you and give you some more information is just to kind of walk you through where to access the information that you need in order to write this unknown lab report. So we are going to start at the calendar in our homepage of our course here. And the first thing that you can do is just click on this assignment, the benchmark identification of an unknown bacterium lab report. And yours will look a little bit different than mine, but ultimately if you click down on this assignment, you'll see an attachment here. And you want to click on that attachment. And this attachment shows you ultimately what you're going to be writing and submitting. So the overall goal of this assignment is to write a lab report that highlights and details the unknown bacteria that you have. And so I'm going to go back to the homepage, but I'm going to keep this tabbed here, and we'll come back to this lab report format guide in just a minute. The next thing that you want to pull up is the assignment in McGraw-Hill. And so I typically find this under syllabus and then class resources and then open up Connect. However you open up McGraw-Hill will be fine. And that will bring you to our class site in the McGraw-Hill Connect server. And then we will go to assignments. And we'll go down to topic 4, which is, not topic 4, topic 5, which is where the unknown bacteria assignment is going to be. And so in McGraw-Hill, then you'll click on topic 5. And we'll go to preview. Once again, your assignment will look a little bit different than mine. You might not have to click through as many things. And ultimately, what you will be pulled to then is an unknown and a number. So here the assignment or the data set that I've been given is unknown number 6. So you can go ahead and open that up. Everybody will have a different unknown. So the number after your unknown is important to include in your lab report, because unknown 6 is going to present different data, for example, than unknown 4. And so when you open up your data set, you can scroll down, and these are experiments that have been done for you. So this is the result of bacterium number 6. You don't know what it is. The goal is to identify what it is. Here's that bacterium on a streak plate of nutrient agar. Here's that bacteria and the way that it grows on McConkey agar. Here's a gram stain of the unknown bacteria. Here is the oxidase test, and you might need to zoom in a little bit to see if there's any result. Here is the UTI-ID membrane test and the eosine methylene blue agar result. So this is all of the results that have been done for you. So this unknown lab report, you're not actually conducting the lab experiments. The lab experiments have already been conducted for you, and your job is to analyze the data, make sense of the data, and then write the lab report. And so this is the data set, and I'll just point out, so unknown number 6 is a different set of data than unknown, that's not unknown, and then unknown number 4. So once again, unknown number 4, it's a different bacterium than unknown number 6. So here's that bacteria streaked out. You can see that it looks different than unknown number 6. Here's how unknown number 4 grows on McConkey agar, and you can see that it grows differently than unknown number 6 does. Why? Because they're different bacteria. So your job is to analyze the data set that you have been given, and that will look different and that will be different for each student. Okay, so that is the data, and I will move that to the side for a minute because there's one more thing that you want to be able to access in order to be successful on this lab report. So we are going to go down now to topic number 5. And on the right-hand side now of topic number 5 is the lab report guide in terms of the instructions for how these experiments have been conducted. So you want to come here, and this will show you how to identify gram-positive bacteria and the tests that are used in that. And then it takes you through test by test. One that we looked at was McConkey agar, which is coming up. You will not have, oh, here's McConkey agar, for example. So you do not have every single one of these tests in your data set, but your job is to look at the tests that have been conducted in your data set, and then come here to these lab experiments and read about each of the procedures that you have been given so that you understand how to interpret them. From that, you can follow one of these two dichotomous keys. So in every data set, there should be a gram stain. If the result of your gram stain is gram negative, then you can come this way. If it's gram positive, you'll come this way. And then you'll follow the test in terms of whether the results are positive or negative for each of these experiments. And that will help you ultimately be able to identify what bacterium you are dealing with. Okay, so this is the page that you're going to want to use to read about the biochemical test, these selective and differential media, to understand what they're doing. This will also be very helpful when you're trying to write your method section. So let's go back now where it talks about the lab report guide. So ultimately, you're going to take your unknown bacteria data set that you get from McGraw-Hill, and then you're going to read about these different tests in the lab manual that we were just looking at. And your goal is to put all of that information into a formal lab report. And what GCU has given you is a guide on how to write that lab report in terms of what needs to be included. First, you will need a title. So your title, as it says here, should be in bold font, centered at the top of the page, and it's telling you to use the same title as the title of the lab exercise. And that title is Identification of an Unknown Bacterium Report. And then you come back here, and here is an example title. Now, the example that they use is for a different experiment than you are writing about. So recognize that it's an example, it's showing you how to do something, but the data and the information that you include is going to be different because you are writing about a different experiment. And so keep that in mind. After you have a title, your lab report should include an aim, which is the main objective of the lab. And so this will be 1 or 2 sentences talking about what your objective is. And keep in mind, the objective of your lab is not to examine the ubiquity of microorganisms in the environment, like the example says. The aim of your lab is to identify an unknown bacterium based on the way that it grows on selective and differential media. And then for the materials, it wants a list of equipment and instruments and cultures and media used, and it's telling you to format using numbers, not letters. So materials, number 1, number 2, number 3, etc. And then you'll go into methods in your own words, explaining what would happen. In order to come up with the materials and methods, you'll want to rely on your lab manual because this is telling you what media is required. And it even includes, if we can go down here a little bit, it even tells you how to do it and what to do. So the directions here, as well as the materials listed here and then the procedure listed here, is ultimately what you'll want to put in your own words in your lab report. And then you come down to results, and here you want to include a data table. Some of this has already been done for you in the unknown data set that you have received from McGraw-Hill. Let me see if I can pull it back up. Oh, here it is. So recognize that this data that you receive from McGraw-Hill is already in a data table. However, it needs a little bit more explanation. And so the lab report guide is asking you to use arrows to label and point out the observations in your results. So you can use the data table from McGraw-Hill as a starting point, but then you want to elaborate on the data table, include additional information, label and identify things that you see. And then in your discussion, this is where you will analyze and interpret the results. So, for example, seeing that these bacteria appear to be gram-negative because they're pink, I would use and discuss that interpretation in the discussion section of the lab report. And then the conclusion kind of ties everything together. So you'll end with a brief paragraph that explains how you accomplished your aim to identify an unknown bacteria using this data. And then importantly, make sure and apply these findings to the healthcare setting. Why is this work important? How is it useful? When you save your file, save it this way. So your full name, underscore, and then the title of the lab, like it says, without spaces and with capitalization. So here's an example here. So that walks you through really how to go about this lab report. You do have 2 weeks to complete this lab report. As I mentioned, you need 3 things. You need your data set that you will get from McGraw-Hill. You want to access the lab manual and the instructions, which you can get from topic 5 and the right-hand side over here, which is right here, this identification of bacteria lab report here. And then you'll also want, which is in the assignment itself as an attachment, right here, you want to have access to the lab report guide when writing your lab report. When you are done, you will upload this entire lab report into Halo. You will upload it. There is a rubric that I encourage you to look at before you submit your report so you know how it will be graded. And this will also require Lopes Write, which checks for plagiarism and lifting material from other sources as well. That is all for this lab report. Let me know if you have questions about it. Good luck.